Welcome to TED Talks Education. We are still for biology. You're with me, Simon. Our main focus in this session is the secondary structure. The secondary structure of a protein. The secondary structure of a protein. We had earlier said that functional proteins take up structures. The first structure is the sequence of amino acids. Now, in this secondary structure, hydrogen bonds package the same polypeptide chain, or chains to form complex structures. In addition to the peptide bonds. So the peptide bonds that are present, those form the primary structure. But when we introduce hydrogen bonds, then we shall be talking about the secondary structure. So we can say hydrogen bonds are formed between adjacent carboxylic group and amino groups. Remember, in our amino acid, we don't have side. So if, let's say, this is the polypeptide chain, that is like that. What is this? A polypeptide chain. And this is exposing the amino group here. We still know that nitrogen is more electronegative, so partial negative, partial positive, partial positive on nitrogen. If in the same chain, the carboxylic group here is also exposing what it has. So this is partial negative, partial positive, partial negative, partial positive. Hydrogen bonds are going to be established, where we are going to see that a hydrogen bond, which we denote with dotted lines, is going to be between hydrogen, which is partial positive, and oxygen, which is partial negative. Even this is going to be established. So this kind of packaging gives us a functional structure which we are calling the secondary structure of a protein. So there are two occurring packages in this. So we can have the alpha helix structure. In the alpha helix structure, you find when one polypeptide chain has been wound to that level. So it's a box, we just call it the alpha helix. Wherever a kink is, you find when there is a hydrogen bond there, packaging it from the alpha helix. Then too, we can have the beta pleated sheets. In this structure, hydrogen bonds come in to join various polypeptide chains together to form what you are naming as beta pleated sheets. So you find something like that. Hydrogen bonds everywhere, uniting several polypeptide chains. Let's say this is chain 1, chain 2, and chain 3. So this functional protein will have 3 polypeptide chains linked together by hydrogen bonds. So those are the two structural arrangements of proteins. The primary structure, which is all about the sequence and kind of amino acids present in the polypeptide chain. Then the secondary structure, which is packaged by hydrogen bonds. So in our subsequent sessions, let's come back and see the tertiary structure and the quaternary